Today, we're tackling, well, maybe the most profound instruction manual there is, the history of the gene.
That's right. It's this tiny, invisible unit that shapes so much of life.
And we've gathered some incredible sources. They trace the idea of the gene from ancient philosophy all the way up to today, where we're actually figuring out how to rewrite the code itself. It covers the discovery, the misuse, the whole story.
Our mission here is really to map that journey. How did we go from just wondering, what is this thing? How are traits passed on?
To the much bigger question now. How do we, responsibly edit it?
Precisely.
Yeah.
And it's not just abstract science.
No, not at all. The source material really brings this home with a personal story. It talks about a family history. Rajesh and Jagu facing this intense volatility, grandiosity.
Right, these phantasms. And the fear that comes with knowing that certain conditions, like bipolar disorder or schizophrenia, can have their risk jump, well, tenfold if multiple family members are affected.
It really underscores why understanding genetic tendency is so critical. It's not destiny, but it sets possibilities.
It sets the stage.
Yeah.
So to really get it, we need to go way back to when people saw the patterns of inheritance, but had zero clue about the how.
Okay, let's dive into that. Ancient times. People knew kids look like their parents, obviously. But how did they think that information traveled?
Well, the early ideas were, let's say, philosophical rather than scientific. You had Pythagoras around the 6th century BC. He had this idea that semen carried all the hereditary instructions gathered from the whole body. Spermism, it was called.
Spermism.
Okay.
And Plato ran with that.
He did, but added this layer of, frankly, social engineering. He proposed a kind of numerological eugenics. He thought if you could just crack the code, the underlying math of heredity.
You could guarantee perfect children for the ruling class. Wow.
Yes, stabilize the republic through biology. It's amazing how quickly that link between knowing and controlling popped up.
But then Aristotle came along. He was more evidence-based, right? He challenged this idea of semen being a traveling library of traits.
Absolutely. Aristotle used logic. He asked, okay, if semen physically contains bits of everything, how does it transmit things that aren't physical? Like a specific way someone walks.
Or traits that don't even exist yet, like a beard on a young boy.
Exactly. How could those things possibly be condensed into a fluid? He argued the mechanism had to be more like instructions, not physical parts.
Okay, so that idea simmers for centuries.
Yeah.
Fast forward to the 19th century, Darwin figures out natural selection, but there's a huge problem.
Well-
A contemporary, Fleming-Jenkin, points it out.
Yes, the blending problem. And it was a serious threat to Darwin's whole theory.
How so? What was the core issue?
Think of it like mixing paint. If heredity always blends traits, say, a beneficial red trait mixed with white, you get pink. Keep mixing generation after generation, and everything just washes out to a murky gray.
So any new advantageous trait, like Jenkin argued, would just get diluted away.
Instantly. It would vanish back into the average. Natural selection couldn't work if variation was constantly being blended out. Darwin himself called it a wide blank in his theory.
So the million-dollar question.
Yeah.
How do you stop the red paint turning gray? And the answer comes from an unlikely place, a monastery garden.
Gregor Mendel. And he was perfect for this because, well, Darwin wasn't much of an experimenter. Mendel's genius was meticulous, hands-on experimentation. Peas. Thousands and thousands of pea plants.
Crossing tall with short round seeds with wrinkled, that sort of thing.
Exactly. Systematically over multiple generations.
Mm.
And what he found blew the blending theory out of the water.
What was the key finding?
Traits were discrete, like little packets of information. When he crossed purebred tall plants with purebred dwarf plants, the first generation wasn't medium height. They were all tall.
All tall. So the dwarf trait just disappeared.
It seemed to. That alone contradicted blending. But here's the crucial part. He then bred that generation together. And in the next generation, the F2 generation, the dwarf trait reappeared, perfectly intact, not weakened or blended at all.
Wow. So it was hidden.
Precisely. And it always reappeared in this very specific ratio. 3 tall plants for every 1 dwarf plant. 3 to 1. It proved heredity worked through these discrete units alleles, we call them now, 1 dominant, 1 recessive.
And the data was famously clean, right? Almost too clean.
So clean that later statisticians actually questioned if he'd, maybe fudged the numbers a bit. The ratios were just textbook perfect across all the different traits he tested.
Incredible. So heredity wasn't blending paint. It was shuffling discrete packets of information. That set the stage for the 20th century quest. What is this packet made of?
Right. And interestingly, a physicist, Erwin Schrödinger, sort of framed the problem theoretically in 1944. He said the gene had to be a molecule of
What
contradictions.
did he mean by that? A contradiction.
It needed two opposing qualities. It had to be incredibly stable and regular, chemically speaking, to copy itself accurately millions of times.
Okay, consistency.
But it also had to be incredibly irregular in its sequence to hold the sheer amount of complex information needed for life, regularity and irregularity in one molecule.
That's a tough design brief. And then Oswald Avery actually nailed down the molecule itself in the 40s.
Yes, he showed definitively that DNA, not protein, as many suspected, was the transforming principle, the actual carrier of genetic information. That discovery kicked off the race to find its structure.
The double helix. Watson, Crick, Wilkins, and Rosalind Franklin.
And our sources really highlight Franklin's crucial contribution. Her X-ray diffraction images, those most beautiful X-ray photographs, were apparently key evidence that Watson and Crick used to figure out that iconic spiral staircase shape.
And the structure itself immediately suggested how it worked, right?
Immediately. That pairing rule A, always with T, G always with C across the two strands, it meant one strand was a template for the other. You could unzip it and make two perfect copies. It solved the information transfer problem Aristotle had puzzled over.
But just as we start understanding the code, the dark side emerges. The desire to control heredity leads to eugenics.
It's a chillingly direct path. Almost as soon as the idea of discrete traits took hold, you see the rise of eugenics, particularly in America in the 1920s. It was fueled by a lot of anxiety about race and immigration.
And this wasn't just theory, it got weirdly practical, like those Better Babies contests.
Unbelievably so. Held at state fairs, like judging livestock. And the scorecards are shocking. Judges were told to give up to 50% of the points based on the baby's heredity, their family background, before even looking at the child's health.
That's horrifying. And it didn't stop there, it became law.
It did. The 1927 Supreme Court case, Buck v. Bell, upheld the legality of involuntary sterilization for people deemed feeble-minded. Justice Oliver Wendell Holmes Jr. wrote that infamous line, "3 generations of imbeciles are enough."
And this American model, it got exported.
And amplified, tragically. The 1933 Nazi sterilization law in Germany explicitly used the American programs as a blueprint, but then ramped it up massively.
And they used propaganda films.
Yes, films like Das Erbe, The Inheritance, and Erbkrank, Hereditarily Ill, showed stark contrast people with disabilities or perceived defects versus the idealized Aryan physique seen in Leni Riefenstahl's Olympia.
It was a deliberate conflation of genetics, identity, and worth, leading to, well, the absolute horrors of the Holocaust.
Exactly. Figures like Joseph Mengele at Auschwitz performing his grotesque experiments on twins were obsessed with unlocking heredity's secrets, using the language of the gene to rationalize genocide. It's the ultimate cautionary tale. Knowing the code creates the temptation to misuse it.
Which brings us forward. After figuring out individual genes, the focus had to shift, didn't it, to understand more complex diseases.
Right. The single gene approach worked for things like cystic fibrosis, but not for the really complex, widespread conditions, things like heart disease, diabetes, or mental illnesses.
Like the schizophrenia that affected James Watson's own son, Rufus. That became a personal motivation for him, didn't it?
It did. Watson realized that schizophrenia isn't caused by one faulty gene, it's a genomic illness. It involves many genes interacting with each other and the environment. Polygenic.
So you couldn't just find the schizophrenia gene. You needed the whole picture.
You needed the entire human genome sequence, a complete reference map. That was the driving idea behind the Human Genome Project, to get that baseline normal sequence to compare against.
And once we had that sequence, that massive book of instructions, the next step was figuring out how it's actually read and used by the cell. Francis Crick's central dogma.
That's the basic flow. DNA holds the master blueprint, it gets transcribed into RNA, which is like a working copy, and that RNA gets translated into proteins, which are the molecules that actually do stuff in the cell. DNA, RNA, Brit protein.
But it's not just a simple reading process, is it? There's regulation involved.
Oh, absolutely crucial. The work of Jacob and Monod showed that genes aren't just coding sequences. They also have regulatory elements attached, like switches, volume controls, punctuation marks.
So it's not just what protein to make, but critically when to make it, where to make it, and how much.
Exactly. These regulatory switches allow for incredible complexity. The same set of genes can be used in different combinations to create all the different cell types and functions in our body.
And that complexity connects back to that idea of tendency we started with. For complex traits, intelligence, personality, risk of illness.
Genetics defines propensity, not certainty. It influences the first derivative of identity, as one source puts it. It sets the initial direction, the potential.
Which leads to that equation the sources highlight. Genotype plus environment plus triggers plus chance equals phenotype.
It's the best summary. Your genes are the starting point, genotype, but your environment, specific triggering events, and just random chance all play a huge role in the final outcome, phenotype, who you actually become. It's not fixed destiny.
Okay, so we understand the complexity, the regulation, the influence of chance. Now, the really big leap, moving from just reading this code to actually writing or editing it.
Yes, the era of gene therapy. And the early attempts were, well, fraught with difficulty. Using viruses to deliver corrected genes proved risky.
The Jesse Gelsinger case in 1999 being the starkest example.
Absolutely. He died during a trial for a metabolic disorder because the viral vector used triggered a massive, fatal immune response. It led to a huge setback, a multi-year pause on many trials. The tools were just too blunt, too unpredictable.
So what changed? What gave us the precision needed?
CRISPR-Cas9. It was a complete game changer. It's like finding a molecular find-and-replace function for the genome.
A copy editing tool for DNA.
That's a great analogy. It allowed scientists to make incredibly precise cuts and edits at specific locations in that 3 billion letter code. You can change a single letter, delete a sequence, insert a new one with remarkable accuracy compared to anything before.
And that precision changes the ethical calculus entirely, doesn't it? Before, interventions were mostly aimed at severe single-gene disorders.
Right. There was this sort of implicit ethical framework. You intervene only when there's a highly penetrant gene causing profound suffering. It was about correction.
But CRISPR opens the door to more.
To enhancement. Because it's so precise, the theoretical possibility arises of not just fixing errors, but adding desirable traits, enhancing normal functions. That possibility caused immediate alarm bells.
Leading figures like Jennifer Doudna called for a pause, a moratorium.
Yes, in 2015, she and David Baltimore, among others, called for a global discussion and caution precisely because they foresaw this slippery slope from therapy to enhancement. They knew the power of the tool they'd helped develop.
And yet, almost immediately.
Almost immediately that same year, a team in China announced they had, in fact, used CRISPR to modify human embryos. Non-viable ones, but the line had been crossed conceptually. We went from theory to practice almost overnight.
So here we are. We've journeyed from Aristotle, wondering about beards, through Mendel's peas, the double helix, the genome sequence, and now we have the tools to edit the instructions themselves.
It's an incredible arc. We now understand genes aren't some rigid blueprint for destiny, but rather these complex, regulated instructions interacting constantly with environment and chance. They shape potential, tendency.
And now we are starting to shape them. Our sources really ponder this next phase. We gain this amazing power to potentially alleviate suffering, maybe even counteract some of the cruelties of chance.
But the risk is losing something fundamental, losing the variation, the unpredictability, maybe even the choices that make us human. If we start correcting everything, what does humanity become?
It closes a kind of logical loop, as the material suggests. If genes determine the organism and we determine the genes,
Then we are determining ourselves in a way never before possible.
which leaves us with that final, powerful thought from our sources to consider. If we start viewing the genome as destiny, does it inevitably lead us to view manipulating it as our manifest destiny? That's the question we face now. The power to write the code is here. What story we choose to write, that's up to us.